What is flow cytometry and leukemia diagnosis? Imagine a powerful tool that can look at thousands of cells in just seconds and tell us exactly what kind they are. That's what flow cytometry does in leukemia diagnosis. It's a laboratory technique that helps doctors understand the characteristics of cells from blood, bone marrow, or tissue samples. The process begins by labeling cells with special fluorescent antibodies. These antibodies stick to specific proteins, called antigens, on or inside the cells. When the sample is run through a flow cytometer, a laser beam shines on the cells as they pass single file. The machine detects the light that bounces off and the fluorescence emitted by the labeled antibodies. This allows the analysis of each cell's size, internal complexity, and which markers it carries. In leukemia diagnosis, flow cytometry is incredibly useful. It helps determine the type of leukemia by identifying whether the abnormal cells come from B cells, T cells, or myeloid cells. For example, if the cells express markers like CD19 or CD20, they are likely from B cells. If they show CD3 or CD2, they are T cells. Markers like CD13 or CD33 point to myeloid cells. This process, called immunophenotyping, is essential for classifying leukemia subtypes such as acute lymphoblastic leukemia or acute myeloid leukemia. It also helps assess how many leukemia cells are present, which is important for staging the disease and planning treatment. Flow cytometry can detect even tiny amounts of leukemia cells, which is vital for monitoring treatment response. This is called assessing minimal residual disease. If the number of leukemia cells drops to very low levels, it suggests the treatment is working well. But if they remains or increase, it might mean the disease is coming back. The technique can analyze thousands of cells quickly, providing detailed information about multiple markers on each cell at once. This makes it faster and more precise than traditional microscope examination. Samples for flow cytometry are usually taken from blood or bone marrow. Bone marrow samples are often used when leukemia cells are not visible in the blood. The process requires fresh, valuable cells and expert interpretation because the data can be complex. Overlap in fluorescent signals can sometimes make analysis tricky, but trained specialists can accurately interpret the results. Overall, flow cytometry is a cornerstone in leukemia diagnosis. It helps identify the specific type and stage of leukemia, guides treatment choices, and allows doctors to monitor how well the disease responds to therapy. It's a highly sensitive and specific tool that complements other tests like genetic analysis and blood counts. If you or someone you know is undergoing testing for leukemia, understanding how flow cytometry works can help make sense of the process. Always remember to consult healthcare professionals for personalized advice and interpretation of test results, as each case is unique.